So this morning it does give me great pleasure to introduce Dr. Wah Chin Boon, who will be presenting our 5th public lecture for the year. Wah Chin is Singaporean by birth, and she completed her doctoral training at the National University of Singapore. I used to live in Singapore, just nearby the NUS campus, and if you haven't been, or if you're planning on going for a holiday, I highly recommend you go and see NUS. It's an absolutely stunning campus. Wah Chin's PhD was, funnily enough, supervised by Professor John Coghlan, who is a name I'm sure is familiar to many of you here. Unfortunately, Professor Coghlan passed away just a few weeks ago, but he obviously made a big impression on Wah Chin, who, following a postdoctoral stint up in Sydney at Prince Henry's Institute, Prince Henry's Institute in Clayton, joined the Florey in 2008. So this actually marks her 10th year at the Florey. Wah Chin heads up the Steroid Neurobiology laboratory here at the Florey, is an NHMRC Senior Research Fellow, and is also supported by the Medical Research Commercialisation Fund. Recently, she also received the inaugural Bio-Autism grant to study animal models of environmental and other molecular interactors with steroid neurodevelopment. And I believe she won't be presenting that work today because it's still in submission at Nature, which I'm sure many of you have also heard of as one of the world's most prestigious medical research journals. So today she'll be talking, giving a more general talk about what we know causes autism and what we have yet to find out. So thank you very much, and please join me in welcoming Wah Chin.
Thank you everyone for coming. And today, I hope the take-home message for everyone is that correlation is not equal to cause and effect. So if you see a correlation of something with a disease, you can't say that it caused the disease until you actually do proper experimentation in animal models to establish that that agent actually caused whatever disease you're interested, autism or cancer. So if you have any question after this talk, you can always email me. Right, so what is autism spectrum disorder? So the interesting thing about autism is the word spectrum. So it's actually, not a homogeneous disease, though not all autistic patients are alike. So each of them have a slightly different deficit, but they all share a core deficit. And these 3 core deficits are the impaired social functioning, impaired language development, and the presentation of repetitive stereotyped behavior. And this includes compulsion or obsession about certain things. Might be some autistic boys might be really interested in dinosaurs, can tell you anything about dinosaurs or cars and stuff like that. So another aspect about autism is there's no blood test. So that we have yet to find a marker in the blood to diagnose a person whether the person has autism or not. So most diagnoses, or all diagnoses, are actually made based on behavior deficit. And there's another quirk about autism is that there's a strong male bias. So there are 4 boys diagnosed with autism compared to 1 girl. So that's why I actually got into the research, because my research focus has been understanding the effects of hormones in the brain. So I thought the best way to understand the effects of hormones in the brain is to focus on a disorder that has a strong male bias, so that I can understand why the brain's, male brains is more susceptible to changes and develop autism. But unfortunately, I won't be presenting those data today. What I will present is data that has been published that show that might contribute to the development of autism. So autism is classified as a neurodevelopmental disorder. That means that the disorder is actually precipitated or induced due to the impairment of the growth and development of the brain or the central nervous system. That means the development of the disease occurs during pregnancy and in the early life stages. Other neurodevelopmental disorders include ADHD. So is there really a rise in autism? So that is actually a controversy. Not all scientists agree on that, because some scientists believe that the rise is due to increased awareness and increased diagnosis. So let's look at the statistics of that. These data are actually found it from the Center for Disease Control in USA. So the first report of autism by the Central Disease Control is in year 2000, and at that time they said that 1 in 150 children has been diagnosed with autism. And in year 2006, they changed the figure to 1 in 110, and then 2010 is 1 in 68. And the most latest statistic they released is 1 in 59. So there seems to have a 100-fold increase since year 2000 in the number of children that have been diagnosed with autism. What's the statistics in Australia? In 2014, it states that 1 in 66 children has been diagnosed with autism. So these data, how do we interpret it? Does it mean that there are more autistic cases, or there are really more children being affected? So are more cases detected, or there are more children being affected? And out of these children that have been diagnosed with autism, only 10 to 20% have been genetically linked. That means that they know what genes have been mutated in these children to precipitate autism. But none of the genes identified so far can actually explain the strong male bias. So does it mean that the other 80% are caused by unknown genes? Or is it because the other 80% are contributed by environmental factors? Right, how do we know that genes actually contribute to autism? So the very first study on autism twins actually conducted in 1977, and this type of study has been replicated again and again. So the common result is that when you look at a pair of identical twins, so identical twins means that they have exactly the same genetic makeup. If one has autism, has been diagnosed with autism, there will be 80% chance of the other one having autism. But in a non-identical twins, or you call it fraternal twins, when one has been diagnosed with autism, there's only 40% chance for the other one to have autism. So that implies that there might be, it's just a very strong evidence that autism is genetically linked. But is there autism genes? So ever since 1977's study, so everyone has been trying to find these autism gene. Only less than 1% of the autistic cases is linked to one single gene mutation. And the list of genes identified or people think that has been associated with autism has been growing, so much so that we actually established a database to collect the data, the reports of all the genes that has been implicated in autism. 65 genes are considered strongly linked to strongly linked to autism, because that means that a quite high percentage of the identified genes mutation belongs to those genes. So one of such genes are SHANK3, another one is Neurobiology, and then there are more than 200 other genes that have weaker ties. That means that some of the people who have those gene mutations do not actually develop autism. So people are confused, like why in some babies have this mutation, they have autism, and then in other babies who have the same mutation did not have autism. That's when the environmental factor might come in play. The common characteristic of all the gene studies is that these genes are either involved in communications between two neurons or controlling the expression of other genes. That means there are transcription factors that actually turn on and off genes in the brain. Yesterday I went to a really interesting talk by the head of neuroscience research in Novartis. Novartis is a big pharmaceutical company. So he actually is really interested in finding the cause of autism. And his mom said to him that son, I'm really glad that you have been making good progress in autism research, because it seems like every single day people uncover a new cause of autism. So it's like, yeah, so it's like every day seems to cause autism. I'll let you have a few seconds to digest this graph. So in this graph, it's plotted of the sale of organic food with the increased incidences of autism. So they do the stats, and the stats here, that means there's a strong correlation. So R equals 1 means it's like a definite correlation, but your logic will tell you that this is not true, right? So correlation doesn't imply cause and effects. So how do we establish cause and effects? One such environmental factors that the scientists have established cause and effects is the maternal immune response. So there were reports that show that there are increase in risk of autism if the mom actually have a flu or have a fever or infection during pregnancy. So what happened in there? So antibodies have been identified as targeted against the fetal brain in 12% of the mothers that have autistic children. And these antibodies were never found to date in children, in mothers that do not have children with autism. Yeah? So this antibody is only found in mothers that have autistic children, 12% of them, and not found in mother of children that develop normally. So to establish the cause and effect, the scientists actually isolate these antibodies from the mom and that have children with autism and inject it into pregnant monkeys. So they inject the antibodies to the pregnant monkeys and see what happened. And sure enough, the offspring, the baby monkeys that's delivered later, actually show altered brain growth. They actually have bigger brains. And they compared to those that haven't been injected with the antibodies, but injected with saline. And they also don't behave normally towards other monkeys or towards the mom. So that one is a concern, that autoimmunity might be an environmental factor that's in utero new word I learned today, in the uterus, the environment in the uterus, that actually affect the brain development. But what causes the autoimmunity is the challenge now. So no one knows what actually triggers the body to actually generate antibodies to attack the fetal brain. So that is one of the areas scientists are working on. And the other factors that has been established, cause and effect, after seeing a correlation is valproic acid. So valproic acid is an anti-epileptic drug. So quite a few studies have shown that children from moms that is taking valproic acid when they were pregnant actually have increased risk. So the biggest study today is this in 2015, where they actually looked at these more than 6,000 children that was born to women with epilepsy. So, but not all the women that have epilepsy, diagnosed with epilepsy, actually take valproic acid, because there are multiple drugs to be, that can treat epilepsy or prevent epilepsy. They prevent seizure. So those children that have been exposed to valproic acid in utero, that means during pregnancy, have the risk of 4.15% of developing autism, whereas the children that was born to the mom that were not exposed to valproic acid, that means the mom did not take valproic acid during the pregnancy, have a risk of 2.44%. So that means that exposure to valproic acid actually nearly doubled the risk of autism. Like I said, correlation is not cause and effect. So how do we establish that the valproic acid actually may increase the risk of autism? Well, so inject valproic acid to pregnant mice or pregnant rats during the 12th day of pregnancy. So what happened is that rat and mice gestation period, that means the period of pregnancy is 21 days, 3 weeks. And 12 days is almost in the middle. And that is the time when the brain is developing. So that is the critical period. So if anything happened during that critical period, the brain will not form properly, and the neurons, the circuit will not connect properly, and that will develop in disrupted circuit. And sure enough, the offspring of these pregnant mice or rats that have been injected with valproic acid actually have decreased social interaction. That means that they do not actually interact with other mice or rats. Mice and rats are actually very sociable. They actually do like to interact with each other. So you know the saying, if you find one mouse or one mouse in the house, there are another 20 for others, because they do not live alone. The other surprising thing or scary thing is the effect is actually transgenerational. That means that if the grandfather has been exposed, or the mom that has been exposed to valproic acid, the grandma and granddad, the grandkids might have autism. So that is so I will show you this data because I find this data most fascinating. So firstly, how do we actually test whether a mouse is autistic or not? So we use a test called a 3-chamber test. So it looks something like that. So they have a box that have 3 chambers, so like 3 rooms. So the middle room actually has have the doorway where the mouse or rat can access to either side. So what we did was we put the scientists, including us, put a mouse into a cage, either one randomly. Then we put the test mouse, and the test mouse will actually, a normal mouse will be curious, they will actually enter the chamber and sniffed out and checked out the stranger mouse. These are the mice that never seen before. So we have the computer program, the recording, to actually record how much time is spent with the mouse sniffing. They can see that they actually try to climb into the cage, to go into the cage to interact with the stranger mouse. And autistic mice do not have this urgency to go and interact with the stranger mouse. So this is the data that I'll walk you through it. So the F1 means the first generation. So the mom of the first generation was injected with valproic acid. So then we wait for the scientists, wait for the mice to be born, and nothing is done to them after they have been born. So the only thing that happened to the F1 generation is that they were exposed to valproic acid in the utero, in the womb. So this is the graph. So this is a measurement of the time. So duration, that means the time spent exploring at the edge of the cage that contains the stranger, or this is the one containing the stranger, or the one that is next to the cage, because we also measure here whether they are sniffing the empty cage itself. So red color are the data from mice that has been exposed to valproic acid. So you can see that it's spending less time compared to a control mice. Control mice are the mice that hasn't been exposed to valproic acid. So this is the first generation. So they, after being exposed to valproic acid, they actually become less sociable. So they actually spend less time trying to interact with the stranger mouse. They took these first generation mice and mated with a naive female mice. Naive means pure, brand new, hasn't been exposed to any chemicals so mice. And then the mom hasn't been exposed to valproic acid, and wait for the second generation to be born. So the second generation is called F2. So remember, F2 hasn't been exposed to valproic acid itself. And you look at this data, so it's quite surprising that they do still F2, that means the children of the F1, valproic exposed mice actually, again, interact less than the control mice, the mice that is from the colony that hasn't been exposed to valproic acid. So that means that the effect of bipolar actually went to the children, which is quite surprising. Then they took F2 and breed it again with another batch of new female mice, and these new female mice hasn't been exposed to valproic acid again, and wait for the F3, the third generation, to be born. So then now this is the grandchildren of F1, and the grandchildren of F1 again is socializing less than the control mice. So what the grandfathers has been exposed to actually pass it down through generations. So this might explain why there is a rise in autism, and might explain why 80% of them, of the cases, autistic cases, we actually can't find what genes is mutated because it's epigenetic changes. But what is epigenetic changes? Epigenetic changes refers to the modification of the DNA that can turn the genes on and off without changing the DNA sequence. So most people realize that the DNA actually encodes for our genetic makeups, and if you mutate the information, that's when trouble happens. You get cancer, you get all sorts of diseases. But in this case here, the sequence itself is not changed. What is changed is that is not on the sequence itself, but on the structure of the DNA. So this is a cartoon. I will walk you through it. So this is the chromosome. So chromosome is actually made out of DNA tightly wound out together around proteins that are called nucleosome. So each chromosome, actually, if you stretch out the DNA length to length, is 5 centimeters. So your DNA for each chromosome is 5 centimeters long. And then we actually have 23 pairs of chromosomes. So if we stretch all the DNA back to back and join up together, it actually will go for 2 to 3 kilometers long. So to me, when I first read this, it's like mind-boggling. So the cells are microscopic, and the DNA is that long. So that makes you understand how the DNA is actually really tightly wound up and coiled up and twisted and packed it up so that it can be stored in the cell. That have a problem. So if the DNA is so tightly twisted and wound up so that it can be packed in the cells, that means that when we need to access the information on these genes, the genes has to be unwound. So little bits of it, the protein will come and unwind a little bit like a ball of wool, so just unwind a little bit of that so that we can access the information. So agents that have epigenetics effect, they will actually lock in. They will actually add molecules to the DNA so that it cannot be unwound anymore. Yeah, so that has been known for quite a while, but the more recent shocking information that this locking in effect is actually passed down through the generations. So even if your grandchildren hasn't been exposed to anything harmful, but because the grandfather had been exposed to harmful substances, it's already done, it's passed down. Yeah, so that is the astonishing thing, is that modification can actually, this epigenetic modification can be inherited. So there are quite a few transgenerational epigenetic agents. This includes pollutants, which were rampant in the 1970s, and people realized that it's dangerous and banned it. One example is this polychlorinated biphenyls. It is actually used as coolant and lubricants in cars, even in electronic equipment. But in 1975, it was found that it's actually quite harmful. It has been banned. But unfortunately, quite a few reports have shown that it is epigenetic agents, and it will modify your DNA, and children of people, children and grandchildren of people who have exposed to these type of pollutants are still suffering the consequence. And other transgenerational epigenetic agents includes pesticides, fungicides, plastics, endocrine disruptors. Endocrine disruptors are chemicals that will interfere with the action of hormones in your body. Yep. So one of the hypotheses that I'm proposing is that the effects of various pollution might be actually accumulating through the generations. That's why you're seeing a rise in autism now. Yep, so we are testing this hypothesis. So in summary, the take-home message today is that autism has multiple causes. Could be genetic, could be epigenetic. Epigenetic is due to environmental factors, and all these interact with each other to impact the growth and development of the brain during pregnancy and early childhood. So some environmental risk factors might actually interact with the genetic factors to produce autism or even alter the severity. So that means that if you actually carry a gene of autism, but you were lucky, your grandparents, your parents, and yourself has never been exposed to environment factors, so you do not develop autism. But if you carry the genes and your grandfather, your grandparents, your parents, or yourself has been exposed to the environmental factors, then they will develop autism, and it might even make the autism more severe. So there might be cases where people do not actually have the autism genes, but because the grandparents, the parents, or yourself have been exposed to the environment factor, so you actually develop a very mild form of autism. So I think it's the interplay between the genes as well as the environment factors that actually produce this spectrum of symptoms as well as the rise in incidences. So. So thank you for your attention. I hope that that's clarified something and you learned something today. And